for this chapter is the cells. We're not going to get through all the cells today. We're going to get through most of them. So I stole the picture from that website that I sent you guys to for Nobel Prize, right? So that means we defend, right? It's the little cells in the little cartoon forms. A couple of them I put in the presentation. So sometimes cute little cartoons help us remember, right? So another thing to help you remember, I found this out of my old workbook for anatomy and physiology class too. Those of you guys taking anatomy and physiology may have seen this. You might, I don't know if you guys are still using this book or not. But there's a saying that we learn, never let monkeys eat bananas. And it helps you remember the order in which the white blood cells are normally in your blood from the most abundant to the least abundant. So N for never stands for neutrophils, which we're going to talk about today. L for let stands for lymphocytes, which we're not going to really talk about too much today. We'll talk about them more when we get to adaptive, because they're more important for our adaptive immune response. M for monkeys stands for monocytes, and we are going to talk about them today. E for eat stands for E for eosinophils, and B for bananas stands for basophils. And one of the things that you'll notice is that 3 of these cells, especially when you look at them under the microscope, and I love looking at these under the microscope, probably my favorite thing to look at. So when they take your blood, they can do a blood smear and they can stain these cells using a special stain. It contains both an acidic and a basic dye. And because of the different components that are inside these cells, they stain different colors, which are somewhat represented in this drawing, but not so well. Neutrophils are going to be the easiest ones to find because, as I said, they're most abundant in your normal blood, right? When you're not experiencing any infection, these are still in relatively high numbers. So they'll make up about 60 to 70% of the white blood cells you would see looking under the microscope. Did I say red blood cells? I'm having a brain fart. White blood cells. I had to say white, right? They're white blood cells. Mostly what you'll see is red blood cells, right? But you're going to be looking around for white blood cells. So these guys are really easy to find. A very key characteristic of them is you'll notice that their nucleus separates into different sections referred to as lobes. And so there are multiple lobes, so they're commonly referred to as being multi-lobed. And so they've got a lot of different names out there. Some people call them polys, P-O-L-Y, for polymorphonuclear, referring to the nucleus has many parts to it. Where most of the cells, right, usually when you're looking at cells under the microscope, you see this, for eukaryotes, our cells, you see this nice big round nucleus, right? So they're distorted in these cells, right, into multiple lobes. And typically they're going to be purple when you stain them using the right stain under the microscope. So that's a pretty characteristic feature when you're looking at them that really kind of stands out. Also in their cytoplasm they have vesicles, commonly referred to as granules. And so they're represented by the little dots here in this drawing. And then you'll notice, so we have neutrophils, we have eosinophils, and we have basophils. And notice they all have these dots, these granules inside of them. So they're collectively known as granulocytes, right? Because they contain granules that stain when we stain them to look at them under the microscope. So they're also named based on that staining. So neutrophils, their granules stain kind of neutral because they're relatively neutral charge-wise. So actually, unless you've really got a really good fresh stain, a lot of times you won't notice the granules inside. right? Sometimes they're kind of orangey, but for the most part, you're usually not going to recognize them. Eosinophils, on the other hand, this is what's really going to help you recognize them, is that their granules stain with the dye eosin, which is an acidic dye. And so they'll range in color from orange to like a reddish color. And they're very distinct, and they'll usually take up most of the cytoplasm. Notice how numerous they are in this picture. Basophils get their name from the fact that they stain with the basic dye, right, that's used in the dye procedure, which is actually methylene blue. And so they come out this bluish purple color. The problem is so is the nucleus, a purplish color. So they tend to kind of blend in together, and it's really hard sometimes to really see the nucleus in these cells. So I have some actual pictures in here that are also in your book from under the microscope that give you a little better idea of the colors. So, but you'll see that these guys are kind of on the bottom of the list, right? If you were to count, say, 100 white blood cells, which is what they do when they do a differential, right, to figure out the percentage, you would only find maybe 2 to 4 of these out of 100 if you're looking at normal blood, someone's not experiencing an infection. Basophils are really rare, right? So you may find 1 out of 100, maybe. Right? So I always tell my students in A&P class when I used to teach that lab, I was like, if you find a basophil, you're going to see Erica very excited do my happy dance, right? Because they're rare, right? The likelihood of actually seeing them in blood. Lymphocytes and monocytes, much easier to find. And for lymphocytes, there's actually 3 divisions of lymphocytes. Natural killer cells, B lymphocytes are sometimes just referred to as B cells, and T lymphocytes sometimes just referred to as T cells. Under the microscope, doing this type of stain, you can't differentiate between those 3. They all look the same. We just call them lymphocytes. They have a nice big purple nucleus and a very little light blue cytoplasm, typically. And then, of course, the biggest ones, which tend to be the 3rd most abundant, and you'll find about 3 to 8 of these per 100 cells you count, are called monocytes. And you see the largest ones, they'll be larger than the red blood cells even. They're really big. And mono in this case referring to the single nucleus, which can be round, but a lot of times it has an indentation in it, so much so that it almost looks like a horseshoe shape, right? So they're characterized by this horseshoe shape. Again, purple nucleus, light blue cytoplasm. And these cells are very large. So some of these cells have the ability to do a process known as phagocytosis. So we refer to them as phagocytes as well. So these are the neutrophils, which are top on our list. And so as you can see here, here's actual picture under the microscope. So you see the multi-lobe nucleus, this purple part. Yep, another ant. Don't crawl on me. And you see the cytoplasm, and you can't even see the granules in this picture, actually. You just see the cytoplasm stained. So that's a neutrophil, as opposed to, and I'll go towards the end of our lecture for the eosinophils. So you see this is the eosinophil. See the multi-lobe nucleus, the purple? But you see that pink-like stuff on the inside? That's all the granules, right, that make that an eosinophil. There's a basophil. Again, you see how it's kind of granular looking? You see these little circles? And you see the nucleus, again, but it's kind of obscure. And I always tell students, notice that all the ones that have granules, what are their names end in? Fill, right? So in this case, literally think of being filled with granules, and then the first part of their name is based on how those granules stain, right? Is it basic staining, right? That blue color for the basophils, or is it eosin staining, the eosin dye, the reddish color? Or is it neutral, your neutrophils, and again, as the name implies, you really don't see their granules because they don't really stain. You guys don't have to be able to recognize a picture of them, but you should know these characteristics, right? That they have the multi-lobe nucleus for the all your granulocytes, and they're all filled with granules. All their names end in "phil." Right? Be able to distinguish one from the other from a description, not necessarily a picture. But maybe I'll be nice if you guys want. I'll put pictures too. It helps. But I wouldn't make you identify it from a picture alone. I would give you a description. Okay. So as it relates to ones that are phagocytes that can do phagocytosis, the first one is our neutrophils. The next is the monocyte. So here's the monocyte. This is a transmission electron micrograph, right? So this is done with an electron microscope. So you can really see detail more than you would see under the light microscope. So you can see that nucleus, right? That in this case, it's kind of horseshoe shaped. And then this is a macrophage. And these two are related to each other. A monocyte is the name given to these cells when they're in your blood, right? So when you see them in your blood, when you do a blood smear, you look at a microscope, it's a monocyte. But these guys travel from the blood into your tissues. When they do that, they differentiate. They become even larger, and they make even more vesicles. So they have vesicles just like neutrophils do, except again, like them, kind of like the neutrophils, they don't really stain well when you look at them under the microscope. So they have some here, but they make even more once they move into the tissues. So you see all these other little dots, right? So here's that really large nucleus, and the angle that we're looking at, it's not that horseshoe shape, but it could be. It just depends where the slice is taken. But again, these are the largest of your white blood cells. So macro, think again large, right? And these are the really big eaters. These guys do a lot of phagocytosis. So what's their job? What is phagocytosis? To clean up, right? They're going to literally engulf and internally destroy foreign invaders. So how are they activated? So remember, part of our system, the very first thing we need to be able to do is if a microbe gets in, what's the first thing we need to do? Or be able to see? Right? So here's this microbe, right? Here's this bacteria cell. It's gotten into our body. How do we know it's there? It's been tagged, right? So we have substances that we produce, things like antibodies, right, that can specifically bind to specific bacteria, right? We haven't talked about the specifics of antibodies yet because that's adaptive immune. That's coming up next. But mannose, right? We have mannose binding lectin. What do we call these things? Right? So what the mannose binding lectin actually binds to on the surface of the phagocyte is a pattern recognition receptor, right? So these are receptors. So we have receptors on those cells to be able to recognize antibodies that have tagged bacteria. So they have what's called an FC receptor. That portion of the antibody the receptor binds onto, what we call the FC region. We have receptors that can recognize C-reactive protein, mannose binding lectin. Complement, remember C3b is what we call an opsonate. It tags it even for destruction, specifically by phagocytes. So through these receptors that these phagocytic cells have, they can recognize, right? They'll activate and be able to phagocytize and destroy these pathogens. So what are cytokines? What are cytokines? So IL-2 is an example of a cytokine, interleukin-2, or interleukin-8. What are those, though, in general terms? So So they're actually grabbing on, right, to these guys. But is that the only thing that happens in our system? When something bad gets in, we just bind to it? How else do we know something is there?
Don't they report to like the other groups coming in?
Right, they report. How do they report? How do they basically, how do they call for help? These molecules, right, the cytokines, right? So we're going to, remember I said the stuff that we talked about was going to keep repeating? Yeah, right? So the cells, not only do they physically bind with receptors, but the cells themselves release cytokines, chemical messages, right, to be able to communicate with each other as to what's going on, right? And that's how these cells become more active, right, go into the area. And you're going to really see that as we look at some of the videos that we're going to look at today. So our two main phagocytes, you can almost say, are our granulocytes, and I stole the picture from that website, and I love it because, look, they put the granules and they look like freckles. So cute. Right? And then, of course, this guy, they drew him really accurately, right? Because macrophages, what do? They eat. So they got this big mouth and these big teeth, right, to really emphasize that these are big eaters. So if we were to compare, and I should have put a title there, shame on me, right? I had my little picture, but it doesn't say neutrophil. If we were to compare our neutrophils to our monocyte macrophages, because we can kind of put these guys in the same category. Add a little bit of space. So neutrophils, as we've said, are the most abundant white blood cells. Right? They're the most abundant you produce even under normal conditions. When you're dealing with infection, their numbers are going to increase. They're going to be the first to arrive during inflammation. Right? So when the cells are damaged, when we have invaders, right, other cells have detected, they're going to release those cytokines, right, and they're going to say, come here. Some of the first ones that are going to get there, because they're so numerous in our blood to begin with, are the neutrophils. And one of the distinguishing characteristics of them, right, is that multi-lobe nucleus you could see in that other picture. They're granules, right? They contain granules, but they're neutral staining, right? So you tend not to see them under the microscope. An example of some of those granules, these vesicles that they have, are specifically referred to as lysosomes because they contain the digestive enzyme lysozyme. What does lysozyme digest? Specifically, peptidoglycan, right? So who's more vulnerable to this, gram-positive or gram-negative bacteria? If it degrades peptidoglycan. Gram-positive, right? Because they have that very thick layer of peptidoglycan. Why not so much our gram-negatives? What do they have that protects their peptidoglycan? Well, it's a thin layer, yeah, but there's another layer outside the peptidoglycan layer for gram-negatives. What did you say, Selma? The outer membrane, right? The outer membrane that contains that LPS that we've talked about, lipopolysaccharide. So it's much harder for the lysozyme to actually get to the peptidoglycan because it has to go through that layer. But, you know, that's, if it's gram-positive, right, we're just going to chew them right up and destroy them. So in addition, you'll notice that macrophages have those as well. They have lysosomes. The difference is that they're referred to as monocytes in our blood. They differentiate into what we call macrophages in our tissues. And in fact, some of them travel to our tissues and stay there as what we call resident macrophages. And so some of them have been given specific names for the different tissues in which they reside in. Right? Because when we were first discovering these cells, right, and we were looking at these cells and say the liver, of course someone named Kupfer named it, right? But come to find out, it's just a macrophage that lives in your liver. So there are lots of different names out there for macrophages that reside in specific tissues, right? So the good news is by the time we got to the lungs, we realized that they were macrophages, and they're called alveolar macrophages, right? But your mesangial cells in the kidney are just resident macrophages. The histocysts in the skin, again, just resident macrophages, right? A more specific name denoting what tissue it's found in. But still the same thing. It's a macrophage. It's an eater, right? Large round nucleus, as we said, or sometimes have that characteristic horseshoe shape. So let's actually watch these guys in action. So this says lymphocyte, and lymphocytes will come into an area during inflammation, but typically it would probably be a neutrophil that is first on the scene. So that's one thing I don't like about is their title to this one, but it is a pretty amazing video. The next is we're going to watch a neutrophil literally chase down a bacteria. So these are videos like I've showed you guys before where it's actually really happening in an organism. Or in the case of the neutrophil chase and the chemotaxis, they've actually taken blood out and put it on a microscope slide and filmed it. But it's the same thing that would happen inside your body. It's pretty amazing stuff. And the last one is chemotaxis. And again, remember the reason why they know to head to a particular area is these cytokines that are released, these chemical messages. Chemo referring to chemical, taxis movement. So hopefully my links work. We'll soon find out. Yay.
To visualize lymphocyte homing to a site of injury.
Okay, so 2 things I need to do. See if the closed captioning works. And we'll find out how accurate it is. Oh, this one doesn't even have it. This one doesn't even have it. Sorry. I think she does put some words up on the screen, though. But what I do need to do is plug it in to the surround sound. And I'm noticing my computer is slow. So I'll do that while I'm at it. So we've already seen how they'll roll out, right? But now they're kind of going to zoom out, and you can see them head in. All right.
A zebrafish larva was anesthetized, and its fin pierced with a needle to introduce a small wound. A vein is seen at the bottom of the frame. Because the fin is very thin and transparent, we can watch directly as lymphocytes crawl out of the blood vessel and migrate towards the wound. They are attracted there by chemicals released from damaged cells, invading bacteria and other lymphocytes. In a zoomed-out view, we can appreciate that lymphocyte invasion is restricted to the wounded area. The static cells that are dispersed in the connective tissue are fibroblasts. In these movies, 60 minutes in real time are compressed into 15 seconds.
Right, so it took an hour for that to happen, right? And they sped this up in a matter of 15 seconds. Yeah, it was a fish larva, so a little tiny baby fish, right? So they poked it with a needle to create this wound that you see here. And then you can see, and I'm going to play it again, but I'll mute her so you don't have to listen to her. I got to pop out. Oh, no, it's right there. Sometimes the symbols just don't make sense to me. All right, so watch. You see them? And again, right, we know they come out of the blood. You see them? Right? And traveling to that site of injury. And when they're zooming out, you can see, as we said, they're just going to that area where they're being called. Right, so remember that these cells themselves that were damaged are going to release chemoattractants, cytokines, that specifically are going to ask the white blood cells to come to that area. Yep, and the mast cells in the area would release the histamine, which actually would cause the vasodilation of that blood vessel, but not not tell them to go to that particular spot, right? That helps with the blood vessel dilation. So the next one, this one's has to be my favorite one, is the neutrophil chase. And no closed captioning either.
Bacteria. Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeking to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
I like the sound effects. I think they're cute. Okay, so this is liquid, right? Because it's actually blood, so it contains a liquid component. Of course, the big things were the these are the red blood cells, right? As I said, here's the neutrophil, and the bacteria, not doing it on purpose, but they release substances normally, and that's also a chemoattractant to the neutrophil that can sense the substances that foreign invaders commonly release. And so he can move just like an amoeba does, right? And he's kind of moving through this liquid media, heading towards. And the only reason why that particular bacteria is moving, because that's a non-motile bacteria, it doesn't have flagella, is because it is a liquid matrix, right? So it's bounced around by thermal energy between the water molecules, right? That's present in the liquid phase of our blood that we call plasma. All right. Yeah, it does seem like it's running, but it's really not. But eventually, right, he catches up with it and engulfs it. So the last one, which is another short one, again, I think really helps drive home the point that those neutrophils know where to go.
These human neutrophils, taken from the blood of the graduate student, are mobile cells that will quickly migrate to sites of interest to help fight infection. They are attracted there by chemical signals that are released by other cells of the immune system or by invading microbes. In this experiment, tiny amounts of chemoattractant are released from a micropipette. When neutrophils sense these compounds, they polarize and move towards the source. When the source of the chemoattractant is moved, the neutrophil immediately sends out a new protrusion, and its cell body reorients towards the new location.
Yep, they move towards the specific chemicals, right? So they actually extracted the chemicals and exposed those cells to them, and then they move just like an amoeba does, right? They push their cytoplasm out, right, to move towards that chemical. So the same thing with the bacteria. It's not doing it on purpose, right? But basically you could say it smells bad, right? And they can sense that, and they're going to head right towards it to be able to engulf them. Now, what these aren't showing is what happens internally, right? So we saw that bacteria end up inside that cell. But what are we doing with it in there, right? And that's the rest of the process of phagocytosis. So let me close this down. Ooh. So I'm not going to spend time on that. There's so many cool videos out there. So you need to be able to describe the steps involved in the process of phagocytosis. So we're going to walk through this. Towards the end of this PowerPoint, I even repeated one of the pictures and took away all the labeling for you. So you can try and label it yourself as you're studying. And then there's another one I want you to label that relates to the next lecture. And then we're going to describe what respiratory burst is and its effect on microbes. And this is one of the things that can happen during phagocytosis, is the respiratory burst. So the first step in the process of phagocytosis is actually being able to bind to the pathogen, whether it be a bacteria, right, a virus, a fungus. So one of the ways that can be accelerated is via opsonization. So specific molecules binding to the pathogen that our phagocytes recognize. So what's an opsonin produced by the complement system that we learned about last time that could be bound to the surface of this, and the phagocytic cell, like a neutrophil or a macrophage or a monocyte, has a receptor for? What's produced? What complement protein is an opsonin? Actually binds to the surface of the bacteria. C for complement. What number? 3. B. Why did I tell you? How do you remember B? Because what does it actually do? It binds, right? Remember B for binding. But other opsonins, right, are antibodies that we're going to learn about. C-reactive protein, mannose binding lectin, right? These things that actually bind to the bacteria and basically tag it for destruction, right? If you think of opsonization, think of it as tagging for destruction. We're marking it. We say this is bad. Now we need to destroy it. And that's what the phagocytic cells will do, our neutrophils, our macrophages, right? So notice it says even here, neutrophil or a macrophage. And what do these guys have inside them? They have lysosomes. So these are vesicles that contain the enzyme lysozyme. So first step, right, actually binding to what it's going to ingest. The next step is it's going to use its cytoplasm, the proteins within it, right, the microtubules and the microfilaments, and it's going to push its plasma membrane around, in this case, the bacteria. In doing so, they're going to create a vesicle on the inside that the bacteria is going to be trapped in. The name we give to that vesicle, because it's through this process of phagocytosis, is called a phagosome. One of the things that happens for this process is that hydrogen ions are going to enter into that phagosome. That's referred to as acidification, right? Because hydrogen ions are acidic, make solutions acidic. The next thing that commonly happens is those lysosomes, right, so those membrane encased vesicles that contain lysozyme will actually fuse with the phagosome, right? So that membrane fuses with the membrane that contains the bacteria in it. In doing so, that lysozyme gets released into that chamber. Because it is a fusion of these two vesicles, they give it a different name. They basically just fuse the two names, right? So instead of just being a phagosome, now when we have the fusion of the lysosome with it, it becomes known as the phagolysosome. So this is when the phagosome containing the bacteria fuses with the lysosome containing the lysozyme enzyme. So that's going to start killing the bacteria, but what if it was a gram-negative we engulfed? Lysozyme's not going to do us too much good, right? So additional enzymes will be pumped into that vesicle, into the lysosome. One of the things that it will pump in, or several of the things, the toxic substances it'll pump in, is reactive forms of oxygen. And this is why it's called respiratory burst, right? Because we need oxygen for respiration, right? Because it's actually accepting electrons for us at the end of the electron transport chain. But oxygen can form toxic forms of oxygen. And these interact with molecules right, and can cause destruction of an organism. So as you can imagine, this is why we keep it encased in a membrane. Because if we just release those things inside the cell itself, we'd kill the cell, our own cell, right, our own white blood cell. So we do this safely within that vesicle, right? So we're just basically keep dumping toxic stuff on whatever it is we engulfed until we kill it. So do you think we always succeed in this? We always kill it and then we just exocytose, fuse that membrane with the outside membrane and throw out the trash? No. There are some pathogens that have evolved that acidification, they can stop that from happening. So guess what? The lysosomes don't fuse. So then they can stay protected within that phagosome and grow. Mycobacteria tuberculosis does that. It will grow inside of our macrophages in our lungs. Yeah, evil little bug. Right. There are others like Shigella. They escape the phagolysosome. They're like, okay, thanks for the free ride, I'm hopping off now. Right? They literally just, they escape. They get out. And then there are some pathogens, this part doesn't happen, right? We don't have any opsonins that can bind to them. We don't recognize them right away. So they don't even get engulfed to begin with. They avoid the process of phagocytosis altogether. So again, although we talk about these protections, there are microbes out there that can get around our system. That's why they can make us sick. Right? They've figured out how to get past our defenses. But this is very effective, right, at killing. So lysozyme, we already know this one, right? This will deteriorate the peptidoglycan in the cell wall, so very effective against gram-positives, not so much against gram-negatives. And that's specific to bacteria, right, because only bacteria have peptidoglycan. Other things they'll release are proteases, and these are enzymes that degrade proteins. So again, we don't want this happening inside of our cell. That's why we do it within the phagosome, right? Why we're going to dump those inside. Defensins, remember we mentioned these before. These are proteins that can insert into microbial membranes. So this would help disrupt that outer membrane of, say, a gram-negative bacteria. Even disrupt the envelope for enveloped viruses. The toxic forms of oxygen that are part of the respiratory burst process are hydrogen peroxide. And so this is something that we sometimes even use, right? Although it's suggested you don't use it on your wounds anymore, because it can be toxic to your own cells, right? If you just dump it on your own cells. You would never want to swallow this, right? It's really bad. You have enzymes to protect yourself against this, right? One of them being catalase that actually breaks it into water and gaseous oxygen, which is why hydrogen peroxide bubbles if you were to put it on a wound. If it doesn't bubble, it means your hydrogen peroxide is probably old and it's mostly water. It should always bubble. You should not, though, they say, put it on your skin anymore. You can rinse your mouth with it, but it's gross. I don't know. I don't know.
Everyone still uses it.
Right. A lot of people probably still use it.
So it mustn't be anything serious.
It's, you know, it's like I said, it's just recommended that it's not your best choice. It's not your best choice. It's not going to hurt you, per se. You know what I'm saying? I think that's probably why, you know, they say big announcements for things that are really going to hurt you as opposed to best practices. So hypochlorite. So this is chlorine ions with hydrogen and oxygen, right? This is basically bleach, y'all. We make it in our cells to kill microorganisms. So hydroxyl radicals, superoxide ions, what they don't show here is it usually has a negative charge to it. And that's why it's so destructive, because it has more electrons than it should, you could say. And nitric oxide. is another one. So these are all toxic forms of oxygen that we can try and throw at microbes in large numbers, so much so even if they have enzymatic protection against it, we can kill them with it. So the last is eosinophils, basophils, and mast cells. So notice I didn't say anything about mast cells yet, right? That's because those typically aren't really found in your blood. These are found in your tissues. They're very similar to basophils, though. You could say they're close relatives. Some scientists believe they're even derived from basophils. So this is another situation where the basophil is in your blood, and then when it goes into your tissue, it differentiates into the mast cells. But mast cells are resident cells in your tissues, right? So these guys don't do phagocytosis. These guys deal with pathogens that are too large to be phagocytosed. But just like neutrophils, right, their name ends in "phil," and they're named after how their granules stain. So what these guys will actually do is they will release those granules outside. So those toxic substances they have bound up in membranes inside, those will go to the cell membrane and be released into the environment, right? So when they come in contact with large things like parasitic worms, right, or when they're activated by allergens, then they're going to release those substances into the area. So that process is called degranulation, because again, we refer to those as granules. So mast cells, right? So notice again, granular. You see the little dots? So here, I can't remember which one they say this is. I think maybe an eosinophil. So these little bubbles you see here are those toxic substances they're releasing. And the green also represents those toxic substances as they disperse. And you can see all the little holes in this poor cell, right? For all those vesicles that came to the surface and released their toxic substances. This is a fungal spore, right, that those substances are attacking. So here is a mast cell. Again, notice all the granules. All these dark circles represent the granules. This lighter mass in here is its nucleus. And you can see them coming to the surface, right, and releasing their substances. The same is true for basophils and eosinophils. They all do this, right? When stimulated, they're going to release these toxic substances into the area. So that's it for today. As I said, try and walk through the steps in this diagram. See if you can label the important parts and steps of phagocytosis. And then you'll notice some of these receptors for some of our cells, I erased them. I want you guys to look in your book and fill them in, because those are going to be important ones that we're going to be talking about. So I want you to be familiar with them for the next lecture. Yes. Maybe. Baria, right? Or Seja. Seja. Selma, you came in late too, huh?
Yeah.
It's not till the end of the semester.
Do we have one test or only test?
Fourth test is on the case studies.
I thought the end of when is the final?
It's December something. It's in Canvas.
What's case study?
I don't have it memorized. So there's a whole module 4. Those are case studies for you guys to go through on your own. And then with your notes, you take the test 4 at home. It's a take-home test.
Oh, okay. Does it affect me in my laboratory? Because I study at home.
Mm-hmm.
That's in this.
Um,
Or it doesn't matter.
I'm just required to take attendance. I don't specifically assign points to attendance.
How does it affect me in a way, like outside this class?
Other than this class, it would be by individual